A very warm welcome to Column Platform, Igniting Curious Mind, where you and I both grow together as on date the 6th June 2026. Biomolecules, there's a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is, are they all living organisms made up of a same chemical elements and the compounds? You have learned in the chemistry how elements analysis is performed. If we had to perform such an analysis on the plant tissues, animal tissues, and microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several other, respective of the content per unit mass of living tissues. In the same analysis is performed on a piece of Earth's crust, as on examples of a non-living matter, we obtain a list. What are the difference between the two lists is that in absolute term, no such difference would be made out. All the elements present in the samples of Earth's crust are also present in the samples of living tissues. However, a close examinations a close examination reveals that a relatively abundance of carbon, hydrogen with respect to other element in the higher in any other living organisms. How to analyze chemical compositions? We can continue asking in the same way what type of organic matter, organic compounds are found in living organisms. How do one go about finding the answers? To get an answer, one has to perform a chemical analysis. We can take any tissues, living tissue, a vegetable or a piece of liver, etc., and grind it in a trichloroacetic acid Cl3CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or a cotton, we would obtain two fractions. One is a filtrate and more technically the acid soluble pool, and the second is a retenant, an acid insoluble fraction. Scientists have found thousands of organic compounds in acid soluble pool. In a higher classes, you will learn how to analyze living tissue samples, identify particular organic compound. It will specific subfig to say one extract of compound, then subject the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolated purifies a compound. Analytical technique when applied to a compound give us an idea of a molecular formula and probable structures of the compounds. All living carbon compounds we get from living tissue can be called biomolecules. However, living organisms also got inorganic elements and compounds in them. How do we know this? A significant different but a destructive experiment has been done. One weighs a small amount of living tissue, say a leaf or liver, and then it is called as a weight. Dry it, all the water evaporates. The remaining materials gives a dry weight. Now, if the tissue is fully burned, all the carbon compounds are oxidized through a gaseous from CO2 and water vapor are removed. What is remained is called as ash. The ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfur, phosphate, etc. are seen in acid soluble fractions. Therefore, element analysis gives the element compositions of living tissues in the form of hydrogen, oxygen, chlorine, carbon, while analysis of the compounds gives an idea of a kinds of organic and inorganic constituents present in the living tissues. From a chemistry point of view, one can identify the functional group like aldehyde, ketone, aromatic compound, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acid, etc. Amino acids are organic compounds containing amino groups and an acidic group such has substitutes as the same compounds. The alpha carbon, hence the called alpha carbon amino acids. The substituted methane, these are four substituent group occupying the four valency positions. These are hydrogen, carbon, carboxyl group, amino group, and variable group designated as R groups. Based on the nature of R group, there are many amino acids. However, those occur in proteins are only of 20 types. The R groups in these proteinaceous amino acids could be hydrogen amino acid is called glycine. A methyl group is alanine and a hydroxyl methyl is serine. The three of the 20 are shown. The chemical and the physical properties of amino acids are essentially of amino, carboxyl, and the R functional groups. Based on numbers of amino and the carboxyl group, there are acidic, that is glutamic acid, basic, glycine, neutral, valid amino acids. Similarly, there are aromatic amino acids, tyrosine, phenylalanine, tryptophan. A particular property of amino acid is ionizable nature of a NH2 and COH groups. Hence, it's in the solutions of different pH, the structures of amino acids change. B is called as a Zwitter ionic form. Lipids are generally water insoluble. They could be simply fatty acids. A fatty acids are carboxylic group attached to R group. R group could be a methyl CH3 or ethyl C2H5 or a higher number of CH2 groups, 1 carbon to 19 carbon. For example, a palmitic acid is 16 carbon, including carboxyl carbon. Arachidonic acids are 20 carbon atoms, including the carboxyl carbon. A fatty acid could be saturated, double bond, without double bond, or unsaturated with one or more C double bond C double bond. Another example, simple lipids, is glycerol. Which is a trihydroxyl propane. Many lipids have both glycerol and fatty acids. Here, fatty acids are found to be esterified with the glycerol. They can be then monoglycerides or diglycerides and triglycerides. They are also called to be fats and oil based on melting points. Oils have lower melting points, that is ginger oil, hence remain as oil in the winter. They can also be identified as fat from the market. Some lipids have a phosphorus and a phosphorylated organic compounds in them. These are phospholipids and they are found in a cell membrane. Lectin is a one example. Some tissues, especially the neural tissues, have lipids with a more complex structure. A living organism has number of carbon compounds in which the heterocyclic ring can be found. Some of these are nitrogenous bases. Adenine, guanine, cytosine, uracil, and thymine are found to be attached to sugar and they are called as nucleosides. In a phosphate group, it's also found to be esterified in sugar and are called as nucleotide. And adenosine, guanosine, thymidine, uridine, and cytidine are the nucleosides. The adenylic acids, the thymelic acids, guanilic acid, and uridylic acid and cytidilic acids are the nucleotides. The nucleic acid, like DNA, RNA, consists of nucleotides only. The DNA RNA functions as a genetic material. A primary and a secondary metabolites. The most exciting aspects of chemistry deals with isolating thousands of compounds, small, big, and from a living organism, determine their structures, and it's impossible to synthesize them. If one were to make a list of biomolecules, a list which would have thousands of organic compounds, including amino acids, sugars, etc. For a reason they are given in the section 9 to 10. We can also call the biomolecules as metabolites. In animals' tissues, one notices that the presence of all such categories of compounds are shown. These primary metabolites, however, are one analysis plant fungal microbial cells. In one would be thousands of compounds, other have these primary metabolites: alkaloid, flavonoids, rubber, essential oil, antibiotics, colored pigments, scents, gums, etc. Spices, these are the secondary metabolites. While the primary metabolites have unidentifiable functions, play a role in the normal physiological process, we do not at the moment understand the rules of other functions of all the secondary metabolites in a host organisms. However, many of them are useful for human welfare: rubber, rocks, spices, scents, and pigments. Some secondary metabolites have ecological importance. In later chapter years you will learn more about this. Some secondary metabolites, pigments, carotenoids, anthracoids, etc., alkaloids, morphine, codeine, and terpenoids, monoterpenes, diterpenes, etc., essential oil, lemon, grass oil, etc. Toxins are racin and lectin are concanvaline A and drugs, vinbastolin and curcumin, etc., and polymeric substances, rubber, gums, and cellulose. Biomolecules. One of the features common to all the compounds found in acidic soluble pools. They are molecular weights ranging from 18 to around 800 Dalton approximately. The acid insoluble fractions has only four types of organic compounds: protein, nucleic acid, polysaccharides, lipids. These classes of compounds are exceptions of lipids, and many molecular weight is ranges from 10,000 Dalton and above. For this every reasons, biomolecules, chemical compounds found in living organisms, are of two types. One, those are molecular weights less than that of 1,000 Dalton and are usually referred to as micromolecules and simply biomolecules, while those which are found in the acid insoluble fractions are called as macromolecules or biomacromolecules. The molecules in insoluble fractions, with the exceptions of lipids, are polymeric substances. Then, why do lipids, those molecular weights do not exceed 800 Dalton, come under acid insoluble fractions? Molecular fractions, lipids are indeed small molecular weight compounds that are present not only as a such but also arranged into a structures like cell membrane and other membrane. When we grind a tissue, we are disrupting the cell structure. The cell membrane and the other membrane are broken into pieces from the vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicle get separated along with the acid insoluble pool. Hence, is a macromolecular fractions. Lipids are not strictly macromolecules. Acid soluble pool represent roughly the cytoplasmic cosmo cosmoposition. The macromolecules from cytoplasm and organelles become the acid insoluble fractions. Together, they represent the entire chemical compositions of living tissues and organisms. In summary, if we represent the chemical composition of living tissue and abundance of points of arrangement, we arrange them in the class wise. We observe the water is the most abundant chemical in the living organisms. Protein are the polypeptide. They are linear chains of amino acids linked by peptide bond. Each protein is a polymer of amino acids, has there is 20 amino acids: alanine, cytine, proline, and tryptophan and lysine. The protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. Thus, information of amino acid content is important as relate to your nutrition lessons. You will learn the certain amino acids are essential for our health and they are to be supplied through our diets. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids are essential and non-essentials. Later are those our body can make. While we get an essential amino acids in our diet and food, a protein carry out many functions in a living organism. Some transport nutrients across the cell membrane, some fight infections organism, some are hormones, and some are enzymes. The some proteins and the functions: proteins, the collagen, intracellular ground substances, the trypsin, that is enzyme, insulin, hormone, antibiotics, fight infectious agents, receptor, sensory receptors, smell, taste, hormone, etc. G gluten 4, that enables glucose transport into the cells. The collagen is the most abundant protein in animal world. Ribulose by bisphosphate, carboxylase, oxygenase, rubisco is the most abundant protein in the whole of the biosphere. Polysaccharides. Acid insoluble pellets are also called as polysaccharides. Carbohydrates has another class of macromolecules. Polysaccharides are long chains of sugars and threads, literally of a cotton threads, containing a typical monosaccharides has a building block. For example, a cellulose is a polymeric polysaccharides consisting of only one type of monosaccharides. Example, that is glucose. A cellulose is a homopolymer. Starch is a variant of this but present in a storehouse of energy and in the plant tissues. Animals have another variant called as glycogen. Insulin is a polymer of fructose. In a polysaccharides chain, says glycogen, the end the right end is called as reducing end and the left end is called as non-reducing end. It has branches as shown in the form of cartoon. A starch forms helical secondary structure. In fact, starch can hold I2 molecules in the helical portion. A starch I2 is a blue in color and cellulose does not contain a complex helix and hence cannot hold a I2. Plants wall are made up of cellulose and a paper made up of plant pulp and carbon fiber in cellulose. They are more complex polysaccharides in the nature. They are building blocks and amino acids and chemically modified sugar. Glucosamine and N-acetylglucosamine, etc., are exoskeletons of arthropods or examples are complex polysaccharides called as chitin. These complex polysaccharides are mostly homopolymer. The nucleic acids are other two types of macromolecules one would find is acid insoluble fractions of living tissues in the nucleic acids. These are polynucleotide. Together, the polysaccharides and polynucleotides are comprises of true macromolecular fractions of any living tissues or cell. For nucleic acid, the building block is nucleotide, and nucleotides has a three chemically distinct components. One is a heterocyclic compound, and the second is homosaccharides, and the third is phosphoric acids and phosphate. As you notice in figure 9.1, heterocyclic compound in nucleic acids are nitrogenous base made up of adenine, guanine, uracil, cytosine, thymine. Adenine guanine is a substitute of purine, while rest is a substitute of pyrimidine. The skeletal heterocycline ring is called as purine and pyrimidine respectively. The sugar is found in polynucleotide as either ribose, that is monosaccharides pentose, or deoxyribose. Nucleic acid contains deoxyribose called as deoxyribonucleic acids, while that contains ribose is called as ribonucleic acid. Structures of protein. As mentioned earlier, the heteropolymer contains a strings of amino acids. Structures of molecules means things in the different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula NaCl, MgCl2, etc. And organic chemistry always written in two-dimensional view of molecular molecules, where representing the structures of molecules benzene and naphthalene, etc. The physics conjure up the three-dimensional view of molecular structure, while the biologists describe the protein structure as a four levels. The sequence of amino acid, the positional information has in a protein, which is the first amino acid, which is second, and so on, called as primary structure of the protein. A protein is imagined to be in a line, and left end represented by a first amino acid, and the right end represent the last amino acids. The first amino acid is called as a N-terminal amino acids, and the last amino acid is called as T-terminal amino acids. Protein thread does not exist through what has extended from the rigid rod. The thread is folded in a form of helix, and of course, only some portion of the threads are arranged in the form of helix. In a protein, one right-handed helix are observed, and the other region of protein threads are folded into a form in which that are called as secondary structure. In addition, long protein chain is folded upon itself in a hollow woolen ball, which give rise to a tertiary structure. The three-dimensional view of the protein tertiary structure is absolutely necessary for many biological activities of a protein. Some proteins are assembling of more than one polypeptide or a subunits. The manner in which these individual flows, polypeptide or a subunits, are arranged with respect to each other. The linear strings of the sphere arranged one upon each other in the form of cube or a plates, etc. In an architecture of a protein, otherwise called as quaternary structure of a protein, the adult human hemoglobin consists of 4 subunits, and 2 of these are identical to each other. The 2 subunits of alpha type and 2 subunits of beta types together constitute the human hemoglobin, Hb. The enzymes, all the enzymes are protein, and these are the nucleic acid that behaves like an enzymes. These are called as riboenzymes. One can depict an enzyme by a line diagram. A enzyme is any protein that has a primary structure, amino acid sequence, and a sequence of protein. An enzyme, like any protein, has the secondary and tertiary structure. When you look at the tertiary structure upon itself, the chain crisscrosses itself, hence many services and the pockets are made. One such pocket is the active sites. An active site of an enzyme is a sieve or a pocket into which the substrate fits. Thus, enzymes that are active sites catalyze reaction at a higher rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at a higher temperature, higher pressure, while enzymes get damaged at high temperature, say around 40 degrees Celsius. However, enzymes isolated from the organisms normally live under extreme high temperature. Hot vents and sulfur spring are stable and retain a catalytic power even at a higher temperature, around 80 to 90 degrees Celsius. The thermal stability is thus an important quantity of enzyme isolation and the thermoacidophilic organisms. The chemical reactions. How do we understand these enzymes? Let's start first understand the chemical reaction. Chemical compound undergo two type of changes: a physical changes and simple refers to change in shapes without breaking bonds. A physical process, another physical process is the change in the states of matter. When ice melts into water, when the water becomes vapor, all these are physical processes, and however, when the bonds are broken and the new bonds are formed during the transformations, this is called as chemical reaction. For example, BaOH2 plus H2SO4 gives BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose in an organic chemical reaction, the rate of physical and chemical process refers to amount of product formed per unit time and is expressed as a rate of delta P by delta T, where the rate cannot be called as velocity if the direction is specified. The rate of physical and chemical process are influenced by a temperature along with the other factor. A general rule of the thumb is that the rate doubles and decreases by half for every 10 degrees Celsius change in the either direction. The catalyzed reactions proceeds at a rate was higher than that of uncatalyzed ones. When the enzymes catalyzed reactions are observed, the rate would be very vastly higher than the same, that is uncatalyzed reactions. The carbon dioxide, water giving you H2CO3, that is a carbonic acid. In the absence of any enzyme, the reaction is very slow, with about 200 molecules H2CO3 being formed in an hour. However, by using enzyme present within the cytoplasm called as carbonic anhydrous, the reaction speeds dramatically with 6 lakh molecules 60,000 molecules being formed every second. Enzymes have been accelerated in the reaction rate by 10 million times. The power of an enzyme is incredibly indeed. The thousands of types of enzymes each catalyze a unique chemical metabolic reactions. A multi-step chemical reactions, when each step is catalyzed by the same enzyme complex or a different, is called as metabolic pathway. For example, glucose to 2-pyruvic acid C6H12O6 plus O2 gives you 2C3H4 plus O3 plus 2H2O. It's actually a metabolic pathway in which a glucose becomes pyruvic acid through a 10 different enzymes catalyzed by metabolic reactions. When you study respiration in class chapter 12, you will study this reaction. At this stage, you will just know that this is very metabolic pathway where one or two additional reactions give rise to a variety of metabolic end products. In our skeletal muscles, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast during the fermentation, the same path leads to a production of ethanol, and hence in a different condition, products of are possible. How do enzymes bring about such high rates of chemical conversations? To understand this, we have to study enzymes a little more. We have already understood the idea of active sites. The chemical or metabolic conversions refers to a reactions. A chemicals which is converted into product is called as substrate. Hence, enzyme. A protein with a three-dimensional structure includes active sites, converts a substrate S into a product P, and symbolically this is called depicted as S to P. Now to understand the substrate, S has bind enzymes at its active sites within a given cleft of or a pocket. Substrate has to diffuse towards active sites. There is thus an obligatory formation of ES Enzyme substrate complex. E stands for enzyme. The complex formation is a transient phenomena. During a state where a substrate is bound to an enzyme active sites, a new structure of a substrate is called as transition state structure is formed. Very soon after the accepted bond breaking, making it completely, the product is released from the active sites. In other words, a structure of a substrate gets transformed into a substrate of product. The path of this transformation must go through so-called transitional states. There could be a many more alternate structure state between the stable substrate and the product. Implicit in this statement in the fact all other are intermediate structural state are unstable. The stability is something related to energy status of the molecules or the structure. Hence, we look at a pictorically through a graph. It looks like a something as in the figure 9.4. Y-axis represent a potential energy and the x-axis represent a progressions of structural transmissions or a state through which a transmissions through the transmission state. You notice that the two thing the energy level differs between the P and S. Energy level, there is a difference in the P and S. If the P is at the lower level than S, a reaction is an exothermic reactions. One need to not supply energy by heat in order to form the product. However, whether it is exothermic or spontaneous reaction or an endothermic reaction or energy required reaction, the S is go through a much higher energy state or a transition state. The difference is in average energy content of S from the other that transmit state. Each enzyme has a substrate binding sites in the molecules who has that the highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociate into a product P and then unchanged enzymes intermediate formations of enzymes product complex. The formation of a EES complex is essential catalyst. The E plus ES gives ES, that is the enzyme substrate, plus EP enzyme product complex that gives you E plus P. The catalytic cycle of enzyme actions can be described in the following step. The first substrate binds to an active site of an enzyme, fitting into an active state site. The binding of substrate induces enzymes to alter its shapes and fitting more tightly around the substrate. The active sites of enzymes are now close proximity of the substrate breaks the chemical bond of a substrate and the new enzyme products of complex is formed. Enzymes releases the product and the reactions and the free enzyme that is ready to bind into another molecule of substrates and run through a catalytic cycle. So once again, factor affecting enzyme activity. An activity of an enzymes can be affected by changing the conditions which can alter the tertiary structure of the protein. This includes temperature, pH change in substrate concentration of binding specific chemicals through a regulator regulates activities. Temperature and pH. Enzyme generally functions in a narrow range of temperature and pH. Each enzyme shows highest activity at a particular temperature and pH called as optimal temperature and optimum pH. Actively decline both below and above the optimum value. A low temperature preserves enzymes in temporary inactive states, whereas in a high temperature destroys enzymatic activity. Proteins are denatured by the heats. The concentrations of a substrate. When an increase in a substrate's concentration, the velocity of an enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, and this which is not exceeded by further rise in the concentrations of the substrate. This is because the enzyme molecules are fewer than the substrate molecules. After such saturations, these molecules, there is no free enzyme molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of specific chemical that bind to an enzyme. When the binding of the chemical shuts of enzyme activity, the process is called inhibitions and the chemical is called as inhibitors. When the inhibitors closely resembles the substrate and the molecular structures and inhibits the activity of enzymes, it's known as a competitive inhibitors. Due to close structural similarity with the substrate, an inhibitor competes with the substrate for the substrate binding sites of the enzyme. Consequence, the substrate cannot bind. As a result, the enzyme action declines. The inhibitions of succinic dehydrogenase by malonate, which closely resembles a substrate, succinate in structure. Such compounds inhibits are often used in the control of a bacterial pathogen. Classification and nomenclatures of enzymes. Thousands of enzymes have been discovered, isolated, studied. Most of these enzymes have been classified in different groups based on their types of reactions they catalyze. Enzymes divide into six classes, each with 4 to 13 subclasses, named accordingly with the four-digit number. Oxidoreductase and dehydrogenase enzymes that catalyze oxidoreductase between the two substrate yes and yes dash, the yes reductase and yes oxidized and yes oxidized plus yes dash reductions. The transferase enzyme that catalyzing the transfer of group G other than the hydrogen between a pair of a substrate and yes dash, yes plus yes G plus yes dash and yes plus yes dash G. The isolated enzymes catalyze the hydrolysis of ester and the ether peptide and glycoside C and helicade and P and N bonds. The lipase enzyme that catalyzes removal of group from a substrate by mechanism other than the hydrolysis, leaving a double bond. Isomerase includes all enzyme catalyzed interconversions of optical geometry and positional isomers. Enzyme catalyzes linking of together the two compounds. Enzymes which catalyze joining carbon, oxygen, carbon yes, and carbon sulfur, carbon nitrogen, and PNO etc. bonds. So cofactor enzymes are composed of one or several polymers polypeptide chain. However, there are a number of cases in which the non-protein constituents called cofactor are bound to the enzymes to make the enzyme catalytically active. This in instances, the protein portions of the enzyme is called the apoenzyme. These three kinds of cofactor can be identified: prosthetic groups, coenzymes, and metal ion. The prosthetic groups are the organic compound that distinguish from the other cofactor in that they are tightly bound to the apoenzymes. For example, the peptide oxidase and the catalyzers, which catalyze the breakdown of hydrogen peroxide to water and oxygen. The HEMA, which is prosthetic group and is a part of an active site of an enzyme. Coenzyme, also the organic compound, and their associations with the apoenzymes are the only transient, usually occurrence during the course of an catalysis. Further, the coenzyme serves as a cofactor in number of different enzymes catalyze reactions. The essential chemical compound of different coenzymes are vitamins. The coenzyme in nicotinamide and adenine and dinucleotide and NADP contains vitamin niacin. A number of enzymes requires metal ion for their activity, which forms the coordination bonds and the side chains, and the active sites at the same time forms one more coordination bonds with the substrate. Zinc is cofactor polyelectrolytic enzymes and carboxypeptidase. The catalytic activity is lost when the cofactor is removed from the enzymes, which justifies that they play a crucial roles in the catalytic activity of the enzymes. Summary: although there is a bewildering diversity of living organisms, their chemicals, composition, and metabolic reactions appears to be remarkably similar. The elements of composition of living tissues and the non-living matters appears almost as to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of a carbon, hydrogen, oxygen is higher in living systems. When compared to inanimate matter, the most abundant chemical in the living organism is water, and there are thousands of the small molecules weigh less than Daltons. 1000 Dalton. The biomolecules, amino acids, monosaccharides and disaccharides, sugars, fatty acids, glycerol and nucleotide and nucleosides, and nitrogen base are some of the organic compounds seen in the living organisms. There are 20 types of amino acids and 5 types of nucleotides. The fats and oils are glycerides, in which a fatty acid are esterified by the glycerol. The phospholipids contain, in addition to phosphorylated nitrogenous compound, only 3 types of macromolecules: protein, nucleic acids, and the polysaccharides are found in the living system. Lipids, because of their association with the membrane, separate in the form of macromolecule or fractions, the biomolecules and the polymers. They are made up of building blocks which are different. The proteins are heteropolymer made up of amino acids and nucleic acids. RNA, DNA are the composed of nucleotide. Macromolecules have a hierarchy of structures: primary, secondary, tertiary, and quaternary. Nucleic acid serves as a genetic material. Polysaccharides are the components of a cell wall. It's a plant, fungal, and also the exoskeletons of arthropods. They also have a strong forms of energy. Example in the starch and the glycogen. Protein serves as a variety of cellular functions. Many of them are enzymes. Some are antibiotics, some are receptors, some are hormones, and some other are structural proteins. Collagen is a most abundant protein in the world. Ribulose bisphosphate carboxylase deoxygenase is the most abundant protein in the whole world of biosphere. The enzymes are proteins which are catalyzed by chemical reactions in the cells. The riboenzymes are the nucleic acids which catalyze the power, and proteinaceous enzymes which exhibit substrate specificity requires optimum temperature pH of maximum activity. They denature at a higher temperature. Enzymes lower activation energy of a reaction, enhance greatly the rate of reactions. The nucleic acid carry the hereditary information and pass from one parental to the next generations. Thank you for watching. You are tuned into Color Platform Igniting Curious Mind, where you and I grow together. Thank you.